Hello, and welcome to this short presentation about enzymes. This is a very important topic in biology, and if you go on to study things like pharmacology or medicine, it'll come up a lot. And I'd say it's one of the key concepts in modern biology. So, to begin with, what is an enzyme? Well, an enzyme is a biological catalyst, and what this means is that the enzyme increases the rate of other reactions without itself being used up. Now, you get catalysts in a lot of different areas, for instance, chemistry, but enzymes exist in living organisms, not just humans, but also animals and plants as well. One of the key things about enzymes is that they are proteins, and this means that the sequence of bases in someone's DNA or an animal's DNA determines the makeup, if you like, of the protein and the enzyme, and therefore how specific it is. So just to draw this out, I'm going to draw a quick cartoon of an enzyme, the structure of it. So here's the protein. And as I said, it's very much a cartoon. And one of the key things about an enzyme is that it has an active site, which I'll draw in a sort of arrow here. So the active site is this area on this side. So for each enzyme, there's one substance or perhaps a group of substances called substrates, which it helps to break down or otherwise increase the rate of their reaction. So these substances are known as substrates. So let's just draw, just changing my colour every now and again. I'll draw the enzyme again. Okay, and then somewhere in the body, let's just say, the enzyme encounters a substrate, which is the substance that it can break down. So this is very much a cartoon representation there. So that's the enzyme and the substrate before they actually bind or associate together. Let's add in another arrow here. The active site of the enzyme, which is where I've labelled on the left there, has a specificity for certain substances or substrates. And when these come into contact, they associate together, which is what I'm drawing right now. And previously, this was known as the lock and key model, the lock and key hypothesis, because the enzyme and the substrate fitted together like a lock and key. But over several years in the late 1950s, a new analogy or theory was put forward, which was called the induced fit theory, whereby the active site actually changed shape a little bit as the substrate came in, and therefore allowed the reaction to take place or speeded up the reaction. So just to summarise what we have so far, enzymes are biological catalysts. They're usually proteins, not all of them, but the vast majority are proteins. They have a specifically shaped active site where their substrate, which I've got in orange here, binds. And this binding or association of the enzyme and the substrate means that the reaction can occur more readily. So as for other catalysts, the rate of reaction increases because the activation energy is lowered, because the substrate is held in a certain way. So what happens next, if I get the pen? So once the chemical reaction has taken place, the products are released from the enzyme. So in this case, it's broken down one molecule into two molecules. And then the enzyme, and particularly the active site, is free again to catalyze another reaction. A key point here for enzymes, as per any other catalyst, is that the enzyme is not used up by this reaction. It's free to go ahead and catalyze other reactions elsewhere. So a few points that I think is really important for you to know about enzymes. So the rate of reaction with enzymes is dependent on quite a few factors. So first of all, temperature. Just put temp for short. As for any chemical reaction, as the temperature increases, the rate of reaction increases. But with enzymes, once you get it beyond a certain temperature, you're actually destroying the three-dimensional structure of the enzyme, and this is known as denaturing it. and therefore the reaction can't take place as quickly. And similarly, the pH has an effect on the rate of reaction as well, for similar reasons, that as the pH gets more extreme, either way, acidic or alkaline, the three-dimensional structure of the protein can no longer exist, and therefore the reaction would be slowed down at extremes of pH. The rate of reaction is also dependent on the concentration of the substrate. So just as a reminder, that's the substance that the enzyme is breaking down. And I've used the square brackets there to indicate concentration. And the other interesting thing about this is that, about enzymes in general, is that you can have competitive and non-competitive inhibitors. So these are molecules which will slow down the reaction. So for the last point, I'm just going to draw the old cartoon of the enzyme one more time. And here comes the substrate. So it's all well and good, the substrate coming into contact with the enzyme, but what happens if, say, something a similar shape to the substrate comes in before it? And this might actually mess up the chances of the substrate going to the active site and undergoing the reaction. So in this case, if a molecule comes in and effectively interrupts the attachment of the substrate, then this is a competitive inhibitor. So the molecule could affect the rate of reaction in other ways, not just by binding on the active site, but also could bind in other places. So similarly, this could also reduce the rate of the reaction, and in this case it would be known as a non-competitive inhibitor. So all of these factors here, let's write in inhibitors. Not brilliant handwriting, but I assure you that says inhibitors. So all of these affect the rate of reaction. We said, one of the key points we said before was that the rate of reaction is dependent on the concentration of the substrate. So let's have a look. If we draw a graph, so we've got our axes there. On the y-axis, we've got the rate of reaction. There we go, the rate. And on the x-axis, let's put the concentration of the substrate. Okay, so the shape of the graph that this would produce, so it starts off fairly straight and then gradually plateaus off until it's pretty much horizontal. So what's happening on the first part of the graph down here, let's just change the colour, change to orange. On the first part of the graph down here, what's happening is that as you get more substrate, it's the same amount of enzyme, more and more of the active sites are getting filled with substrate, so the rate of reaction is going up in a linear fashion, in other words, a straight line. But then as you get up to this section here, what's happening is that more and more of the active sites of the enzymes are actually getting saturated, so there's none of the active sites left to catalyze the reaction, and therefore you're getting this kind of plateau effect where there are actually no more active sites left. And this is a very important graph, as if you study perhaps pharmacology, you'll go into this in more detail. So I hope that's a nice introduction to enzymes for your studies. We'll go back to the original little drawing there. So in summary, enzymes are biological catalysts. Most of them are proteins. They have a very specific structure which comes from their three-dimensional or tertiary structure, and a very key concept is their active site, which you can see the kind of wavy lines here. This isn't necessarily exactly the same as the substrate here in orange, but it certainly has a complementary shape, and the specificity does vary between different enzymes and different substrates, but the key point is that they're similar enough to allow the rate of reaction to speed up. So thanks very much for listening, and I hope that was useful.